Welcome to Kalang Platform Igniting the Curious Mind. Let's begin the chapter. The plant growth and development. You have already studied the organizations of flowering plant in chapter 5. You have thought that they about where and how the structures like roots, stems, leaves, flowers, seeds arises and what is to ordinary sequences. You are by now aware of the term seed, seedling, plantlet and mature plants. You have seen that trees continues to increase in heights and growth over a period of time. However, the leaves and flowers and fruits of the same tree not only have a limited dimensions but also appear fall periodically, sometimes repeatedly. Why does a vegetative phase precede flowering in plants? At the all plants organ made up of varieties of tissues. Is there are any relationship between the structures of a cells, a tissue, an organ, and the function they perform? They can the structure and the functions of these be altered? All the cells of a plant are descendant of zygotes. The question is then why and how are they have different structure and functional attributes? Development is a sum total of the process, growth, and differentiations. To begin with, it's essential and sufficient to know the development of a mature plant from a zygote fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that proceeds roots, stems, leaves, branch, flowers, fruits, and seeds, which eventually die. The first step in the process of plant growth is seed germination. The seed germination, when favorable condition growth exists in the environment. In the absence of unfavorable condition, the seeds do not germinate and does not produce thus goes into a period of suspended growth or the rest. Once a favorable condition returns, the seeds resumes metabolic activity and growth takes place. In this chapter, you shall study some of the factors which governs and controls the developmental process. These factors are both intrinsic, internal, and extrinsic, that is external to the plant. The germination of seedling development in wheat. The Growth is regarded as one of the most fundamental and conspicuous characteristics of living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its parts or even the individual cells. Generally, growth is accompanied by metabolic process, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. What would how you describe a swelling of a piece of wood when you placed in the water? Plant growth is generally interdeterminate. Growth is a unique because plants retain capacity for unlimited growth throughout their life. This ability of a plant is due to the presence of meristematic at certain location of their body. The cells of such meristem have the capacity to divide, self-perpetuate. The product, however, soon loses the capacity to divide, and such cells can make a plant body. This form of growth, wherein a few cells are always being added to the plant body, is by the activity of meristem is called open form of growth. What would happen if a meristem ceases to divide? Does this ever happen? In the early classes, you have studied about the root apical meristem, shoot apical meristem. They are also responsible for the primary growth of a plant. Principally contributes to the elongation of the plants along their axis. You have known that in dicotyledonous plants and gymnosperm, in the lateral meristem, the vascular cambium and the cockambium appear later in life. These are the meristems that causes an increase in the growth of an organ in which they are active. This is known as secondary growth of a plant. Growth is measurable at a cellular level, principally a consequences of increase in the amounts of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantitative which is more or less proportional to it. Growth is therefore measured by the variety of parameters, some of which are increase in the fresh weight, dry weight, length, and area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem gave rise to more than 17,500 new cells per hour, whereas as the watermelon can increase in size up to 350,000 times. In former, growth is an expression of increase in number cell. cell number, and later express growth in an increase in size of the cell. While the growth in the pollen tube is measured in terms of length, an increase in surface area denotes the growth of dorsiventral leaf. Phases of growth. A period of growth is generally divided into three phases, mainly meristematic, elongation, and maturations. Let us understand this by looking at a root tip. The constantly dividing cells, both at the root apex and shoot apex, represent the meristematic phase of growth. The cells in these regions are rich in protoplasm, possess large conspicuous nuclei. The cell walls are primary in nature, thin and cellulosic with abundant plasmodesmata connections. The cells are proximal, just next away from the tip of the meristematic zones, represent the phase of elongation. Increased vacuolizations and cell enlargement, cell wall depositions are the characteristic of cells in these phases. Further away from the apex, that is more proximal to the phase of elongations, lies the portion of axis which is undergoing the phase of maturations. The cells of the zones attain their maximum size in terms of their wall thickening, protoplasmic modification. Most of the tissues and cell types you have studied earlier in the class represent these phases. Growth rated. The increase per unit time is termed as growth rate. The rate of growth can be expressed mathematically. An organism or a part of the organism can produce more cells in variety of ways. Arithmetic, geometric, and zygote divides. The geometric phase cell divides cells. Arithmetic phase, diagrammatic representation of arithmetic and geometric growth. The stage during the embryo development showing geometric and arithmetic phases. Growth rate shows an increase that may be arithmetic or geometrical. In arithmetic growth, the following the mitotic cell divisions, only one daughter cell continues to divide while the other differentiates and mature. The simplest expression of arithmetic growth is that they elongate by root elongation at a constant rate. Look at the figure. On plotting the length of an organ against the time, and a linear curve is obtained. LT is equal to L0 by RT, where LT is the length of it at the time t and the L0 is the length of a time at 0, a growth rate and per elongation per time. Let us now see what happens if a geometric growth. In most of the time, initial growth is slow, that is lag, and it increases rapidly thereafter at an exponential rate. A log or exponential phases. Here, the progeny cells following the mitotic cell divisions retain the ability to divide and continue to do so. However, with a limiting nutrient supply, the growth slowed down, leading to a stationary phase. If we to plot a parameter growth against time, we would typically have a sigmoid curve. A sigmoid curve is a characteristic of a living organism growing in natural environment. It's a typical of all cells, tissues, and organ of a plant. Can you think of some similar example? What kind of cone can you expect in tree showing seasonal activities? Now, exponential growth can be expressed as a W0 by ERT, where F1 is a final size, weight, height, or number. W0 is initial size of the being at the period, and R is for growth rate, TE for time of the growth, E for base of the natural logarithms. R is a relative growth rate and is also measured in the ability of the plant to produce a new plant material, refers to as efficiency index. Hence, the final size of W1 depends on the initial size W0. The quantitative comparison between growth of living system can be also made in two ways. The measurement and the comparison of total growth per unit time is called as absolute growth rate. The growth rate of a given system per unit time expressed on a common basis. Per unit time initial parameter is called relative growth rate. The two leaves A and B are drawn that of area different sizes but shown absolute increases in the area in a given time to a given leaves A1 and B1. However, one of shows a much higher relative growth rate, which one and why. The conditions of a growth. Why do we not try to write down what do you think are necessary conditions for a growth? Let's list out list have water, oxygen, nutrition has essential elements of growth. The plant cells grows in size by enlargement, which in turn requires water. Turgidity of the cells helps in extension's growth. This water growth and the further development in intimately linked to the water status of the plant. Water also provides a medium of enzymatic activities needs for growth. Oxygen helps in releasing metabolic energy essential for growth activity. Nutrients, macro and micro essential elements are required by plants by the synthesis of protoplasm acts as a source of energy. In addition, every plant organism have optimum temperature or rather it suits for their growth. Any deviation from the range could be detrimental to the survival. Environmental signals such as light and gravity often also affect certain phases and stages of a growth. Differentiation, redifferentiation, and redifferentiations. The cell divide from the root apical and shoot apical meristem, and the cambium differentiate by mature to perform a specific functions. This acts as leading to a maturation in terms of differentiations. During differentiation, cells undergo a few major structural changes both in their cell wall and the protoplasm. For example, to some of the tracheiry elements, the cells would lose their protoplasm. They also develop a very strong elastic and linocellulosic secondary cell wall to carry water to a long distance even under extreme tensions. Try to correlate the various anatomical feature you encounter in the plants to the functions they perform. A plant show interesting phenomena. The different living differentiated cells are now have lost the capacity to divide and regain the capacity to divide under certain conditions. This phenomena is termed as de-differentiation. For example, formation of meristems in the intravascular cambium, the cocambium, which fully differentiated parenchyma cells. While doing so, such meristem and tissues are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific functions, that is redifferentiations. List some of the tissues in Audi dicotyledonous plants that are the products of the redifferentiations. How could you describe tumor? What would you call the parenchyma? Cells that are made up of divides made to divide under controlled laboratory conditions during the plant tissue culture. We have mentioned that the growth in plants is open. It can be intermediate or determinate. Now we may say that even differentiation in the plants is open because cells and tissues arises out of same meristems have different structures at maturity. A final structure at maturity of a cells and tissues is also determined by location of the cells within. For example, cells positions away from the root apical meristem differentiate at a root cap cells, while those pushed to the peripheral mature at epidemals. Can you add more examples of open differentiation correlating the opposition to cells, the position in an origin? Development is a term that includes all the changes an organism goes through during its life cycle for germinations of seeds in acinesis, a diagrammatic representation of sequence of process which is constitutes the development of cells of a higher plant in the figure. It's also applicable to tissues and organ. Plants follow a different pathway in response to environment or the phases of life to form a different kinds of structure. This ability is plasticity. The heterophyly is the cotton, coriander, and the larkspur is in such plants. The leaves of a juvenile plants are different from the shapes that they are in nature. Mature plants on the other hand, differences in shapes of leaves produces in air and those produced in water in a buttercup also represent the heterophilous development due to environment. This phenomena of heterophyly is example of plasticity. Thus, growth is differentiations and development, also a very close related event in the life of a plant. Broadly, development is considered as the sum total of differentiations. Development in plants, both growth and differentiation, is under the control of intrinsic and extrinsic factor. The former includes both intracellular genetics and intracellular factors, the chemicals such as plant growth regulator, while the latter includes light, temperature, water, oxygen, nutrition, etc. Plant growth regulator characteristics. The plant growth regulators are small simple molecules of diverse chemical compositions. They could be indole compound, indole triacetic acid, adenine derivative CN6 furfuryl amino purine kinetin derivatives of carotenoid abscisic acids ABBA terpenes, that is glyceric acids, and GA3, and gases ethylene C2H4. The plant growth regulators are variously described as plant growth substances, the plant hormones, or phytohormones in literature. The PRG can be broadly divided into two groups based on their functions in the living body. One kind of PRG are involved in the growth promoting activities such as cell divisions and cell division elongation patterns of formations, the growth tropic growth, flowering, and the fruiting and seed formation. These are the also called as plant growth promoters: auxins, gibberellins, and cytokinin. The plant PRGs, the plant growth growth regulators, or other groups which may have important role in the plant response to wounds and stress and biotic and abiotic origins. These are involved in various growth inhibitor activities such as dormancy and abscission. The plant growth regulator abscisic acid belongs to this group. The gaseous plant growth regulator ethylene out could fit either by a group, but it's also largely by inhibitor of growth activity. The discovery of plant growth regulators. Interestingly, the discovery of each of these five major groups of PGRs have been accidental. All these started with the observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptiles of a canary grasses responds to unilaterally illuminations by a growing towards a light source, phototropism. After seeing a series of experiments, it concluded that the tip of the coleoptile was a site of transmissible influences that causes the bending of the entire coleoptile. The auxin was isolated from EFW went from the tip of the coleoptile of an oat seedling. The bannakin, that is foolishing seeds, a disease of a rice seedling, was caused by the fungal pathogen Gibberellin fugicori. The Kuroswa reported an appearance of symptoms of disease in the rice seedling, and then they treated with a sterile filtrates of fungus. The active substance was later identified as gibberellic acids. The F. Skoog and his co-worker observed that the internodal segments of tobacco stem, the callus, a mass of undifferentiated cells, proliferated only if the additions of auxin in the nutrient medium was supplemented with one of the following: extracts of vascular tissues, yeast extract, coconut milk, or DNA. The Miller et al. 1955 later identified and crystallized the cytokinins promoting active substance that were termed as chitin. During mid-1960, the three independent research reporters at the purification and the chemical characterization of three different kinds of inhibitors, inhibitor B, abscission through, and dormin. Later on, the three were proved to be chemically identical. It was named as abscisic acids, ABA. The H.S. Carson 1910 confirmed the release of volatile substance from the ripened orange and Haston, the ripened stored unripened bananas. Later, this volatility substance was identified as ethylene and a gaseous PGR. Let us study some of the physiological effects of these five categories of PGAs in the next sections. The physiological effects of plant growth regulators. Auxin. Auxin, from a Greek word auxin, to grow, was first isolated from a human urine. The term auxin is applied to iodine 3 acetic acids, IAA, and to the natural and the synthetic compounds of certain growth regulator properties. They are generally produced by a growing apex of a stem and a root, from where they are migrated to a regions of their actions. Auxin, like IAA, and the indole butyric acid IBA was being isolated from the plants. The NNA naphthalene acetic acid and 2,4-D diaceto 2,4-D, that is 2,4-D dichlorophenol acetic are synthetic auxin. All these Auxin have been used extensively in agriculture and horticulture practice to help to initiate the rooting in the stem cutting and application widely used in the plant propagation. Auxin promotes flowering in pineapple. They help in prevent fruits and leaf drop at early stage but promotes the abscission of the older mature leaves and fruits. In most higher plants, the growing apical buds inhibits the growth of a lateral axillary buds, a phenomena called as apical dormancy. The removal of shoot tip decaptation usually results in the growth of nat lateral buds. It's widely applied in the tea plantations and hedge making. Can you explain why auxin is induced by a parthenocarpic, that is by tomato? In the tomato, they are widely used in herbicides to 4D and widely used to kill dicotyledonous weeds. Does not affect mature monocotyledonous plants. It's used in prepare the weed-free lawns of gardens. Auxins also controls the xylem differentiation, helps in cell division. Gibberellin. Gibberellin is another kind of promotion of PGR and are high and more. There are more than thousands, hundred gibberellin reported widely different organisms such as fungi, higher plants. They are denoted as GA1, GA2, GA3, and so on. However, gibberellic acids is one of the first gibberellin to be discovered and remain the most intensive studied all from. All GAP are acidic and they are produced in a wide range of physiological response in the plants. Their ability to cause an increase in length of auxin is used to increase the length of a grape stalks. The gibberellin causes a fruits like apple to elongate and improve its shape. They are delay senses. Thus, the fruit can be left on a tree for longer so as to extend the market period. The GA3 is used to speed up the malating process in the beverage industry. Sugarcane stores carbohydrates as sugar in their stems. Spraying of sugar crops and gibberellin increase the length of a stem and thus increases the yield of such 20 tons of per acre. The spraying of a gibberellin confers a GA3 hastily maturity period and thus leading to an early seed production. Gibberellin also promotes blotting internode elongation and this prior to flowering in beets and cabbage and many other plants and thus rosette habits. Cytokinins. Cytokinins have a specific effect on cytokinesis were discovered as chitin, a modified form of adenine purine. The autoclave hearing sperm DNA. The chitin does not occur naturally in the plants. Search of these natural substances are cytokinins activity lead to the isolation of zeatin from the corn kernel and the coconut milk. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with the cell division promotes activity which have been identified. Natural cytokines are synthesized in regions a rapid cell divisions occurs. For example, root apex development, shoot buds and fruiting and young fruits. It helps in produce new leaves and chloroplast in the leaves. Later, shoots growth and adventitious shoots formation. Cytokines helps in overcome the apical dormancy. They promote nutrition metabolism. It helps in the delay of the leaf sensors. Ethylene. Ethylene is a simple gaseous PGR which is synthesized in a large amount by a tissue undergoing senescence and ripening of a fruit. Influences the ethylene on the plants and includes horizontal growth of the seedling, the swelling of the axis and apical hook of formations in dicot seeds. Ethylene promotes senescence and abscissions of the plant organs, especially leaves and flowers. Ethylene is highly effective in a fruit ripening. It enhances the respiration rate during ripeness of the fruit and this raises the rate of respiration called as respiratory climatic. The ethylene breaks seeds and bud dormancy, initiate germinations in the peanut seeds, sprouts of food potato tuber. Ethylene promotes rapid internode petiole elongations in a deep water rice plants. It helps the leaves to upper plant upper part of the shoots to remain above water. Ethylene also promotes root growth, root hair formation, thus helping the plant to increase their absorption surface. Ethylene is used to initiate flowering and also synchronize fruit sets in pineapple. It also induces the flowering in mango. Since ethylene regulates so many physiological processes, it's one of the most widely used PRG in agriculture. The most widely used compounds of the source of ethylene is ethifon. Ethifon is an aqueous solution which is readily absorbed and transported using within the plant and release ethylene slowly. The ethifon fastens the food ripening in tomatoes and apples and accelerates the abscissions in the flowers and fruits. 3 thinning the cotton and the cherry walnut. It promotes a female flower in cucumber, thereby felling off it promotes the female flowers in the cucumbers, thereby increase the yields. Abscisic acids. As mentioned earlier, abscisic acids was discovered for its role in the regulations of abscission and dormancy. But like other PRGs, it has been a other wide-ranging effects of plant growth development. It acts as a general plant growth inhibitors and inhibitor of plant metabolism. ABBA inhibitors seeds germination. ABA, that is, abscisic acid, stimulates the exposure of stomata, increases the tolerance of the plants to various kinds of stress, and therefore it's called as stress hormones. Abscisic acids plays an important role in the seed development, maturation, dormancy. By inducing dormancy, abscisic acid helps seeds to withstand desiccation and other factors unfavorable for the growth. In most situations, abscisic acid acts as an antagonistic of GA. We say that the Gibberellins. We say the summarize that we may summarize that any other every phases of growth, differentiation and development of plants, one or the other PGAs has been role to play. Such role could be a complementary or antagonistics, and these can be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where there are more than one PGA interacts with to affect the events. Dormancy in seeds, buds, abscissions, and senescence in apical dormancy, etc. Remember, role of PAJ is the only one kind of intrinsic control. Along with the genomic control and extrinsic factor, they play an important role in the plant growth development. Many other extrinsic factors such as temperature, light control plant growth development via PGA. Some of these events could be a vernalizations, flowering, dormancy, seed germination, plant movement. We shall also discuss briefly the role of light and temperature. Both of them has extrinsic factor on the initiation of flowering. Summary: growth of one of the most conspicuous events in any living organism. Its irreversible increase in expression of parameters such as size, area, length, height, volume, cell number, etc. Inconspicuous involves increased protoplasmic material. In plants, meristematics are the sites of growth. Root and shoot apical meristem, sometimes along with the intercalary meristem, contribute for elongations of the plant apex. Growth is intermediated cells. The growth could be arithmetic or geometrical. Growth may not be general or sustained to a high rate throughout the life of a cell, tissue, organ, or organism. Can be defined three principal phases of growth: a lag, log, and a senescence phase. When the cell loses the capacity to divide, it leads to differentiation. Differentiation results in development of structures that is compensates with the functions of the cells which finally have to perform. The general principle for differentiation of a cell, tissue, organs are similar. Differentiation cells are dedifferentiated and then redifferentiated. Since the differentiated in the plant is open, development could also be flexible. The development in the sum total of a growth and differentiation. The plant exhibit a plasticity in the development. Plant growth and development are under the control of intrinsic and extrinsic factor. Intercellular intrinsic factor are the chemical substance called as plant growth regulators. These are diverse group of PGA. The plants are principally belonging to 5 groups: auxin, libric acid, cytokinin, abscisic acids, ethylene. These PGAs are synthesized in the various parts of plant. They control the different differentiation and development event. The PGAs have a diverse physiological effects on plants. The diverse PGA also manifests a similar effect. The PGA can act as a synergistically and antagonistically. The plant growth and development is also affected by light, temperature, nutrient, oxygen status, and gravity, such as external factor. Thank you for watching. You're tuned in.